Hello and welcome to this virtual open day. My name is James Hodgkinson and I'm a lecturer here at the University of Leicester. And I've been asked to give you a brief research talk on the type of research I do here at the University of Leicester. Now, there may be a lot of terms here that you maybe don't understand or unfamiliar with, but don't worry about that too much. I'm just trying to give you a flavour of what I do. And the piece of research I'm going to talk about today is chemical tools for probing histone deacetylase multiprotein complexes in disease. So my main research is in the synthesis of novel molecules to study biological systems. So I'm a chemist and I'm a synthetic chemist and a medicinal chemist, but I like to make molecules for biological applications. There's two areas of research that I'm really interested in. One is in bacterial iron transport, which is important in antibiotic development. And the other, which I'm going to talk about in this short research presentation, is histone deacetylase enzymes, abbreviated as HDACs. And I'll talk a lot about these enzymes in the coming presentation. Okay, so researchers don't normally work in isolation. I work as part of a collaborative team. So this is me speaking now, Dr. James Hodgkinson, and I'm a chemist by training. And I do the synthesis and the chemistry of these HDAC modulating compounds. However, this is a multidisciplinary research project, and I work in collaboration with Professor Sean Cowley, who works on the cell biology of HDAC complexes, is also at the University of Leicester, and he will use my molecules in his cancer cells to see what they do. And Professor John Schwabe, who is a structural biologist of HDAC complexes, and he gets images and snapshots of how these HDAC proteins interact with the molecules that I make in my lab. Okay, so why probe histone deacetylase complexes? Basically, in every human, there are 18 HDAC enzymes. 11 of those have zinc in their catalytic active site. And not only that, these class 1 HDACs, so 1, 2, and 3, exist in several multiprotein complexes. So there are quite a few HDAC enzymes in the body. And they exist also in different types of multi-protein complexes. So what they do is in the nucleosome, in your genes and your DNA, you have proteins wrapped around them, and they will simply remove an acetyl group from these proteins. Now that may sound very simple, but that determines what genes get turned on or off in the body. And HDAC inhibitors are known drugs that have therapeutic benefits in a number of diseases. So this is forinostat, which is a HDAC inhibitor drug that's used to treat many cancer patients in the clinic. However, there is a problem with forinostat. Although it can be used as an anti-cancer agent, it has nasty side effects. And one hypothesis is that forinostat is indiscriminate. It inhibits all of the 18 HDAC enzymes. So what we would like to do is make molecules that are specific to specific HDACs. So if we can make molecules to engage a HDAC with complex selectivity and enzyme selectivity, we can selectively probe the function and phenotype related to a specific HDAC complex in the cell. And we can determine the relevance of certain complexes to roles in disease in differing cancers and differing neurodegenerative disorders. So there may be one particular HDAC enzyme or complex that's particularly important in a certain type of cancer that we can design small molecules to work. And it will also provide chemical scaffolds for starting points for drug discovery. So one day, maybe these molecules could be used in the clinic to treat patients with cancer or neurodegenerative diseases. So this is the other term I want to talk about. So we want to do this by a special way. So we use these molecules called PROTACs. Now don't worry too much about that abbreviation, but basically they're heterobifunctional molecules. So molecules that consist of two components. They have a ligand for the protein of interest. They have, well, three components: a linker and an E3 ligand. Now, don't worry too much if you don't understand those concepts. But this would be your PROTAC here. So we have the ligand for their HDAC complex, we have a linker, and we have a ligand that recognize this E3 ligand. Now, this part of the molecule utilizes the proteasome of the body to degrade the proteins of interest. So that's what's really novel about PROTACs. Most drugs inhibit a catalytic active site. PROTACs don't do that. They degrade the protein of interest. And there's a lot of excitement about these PROTACs because they have demonstrated vastly improved selectivity and potency compared to drugs such as vorinostat, which I just talked about. And the idea is, can we use PROTACs, sorry, there's a spelling mistake there, to degrade HDACs with multiprotein complex selectivity? So this hasn't been investigated before. This is our really area of research. And what would happen is we'd have our HDAC, which has many proteins around it. We have our PROTAC and our linker E3 ligand, which will bind our HDAC complex. So we have a binding event between these two. Then, because we have this E3 ligand here, it will use the proteasome to make a protein-protein interaction between this E3 protein and the HDAC protein. And that will result in the HDAC being degraded. And if the HDAC is degraded, disappears, then we will regenerate our PROTAC and we'll have a catalytic cycle. So we can have a very potent molecule. Again, that's very different to normal drugs that are used in the clinic. Most drugs just inhibit protein function, where we don't want to just inhibit the protein function. We want to completely remove the protein and completely degrade it for an even greater therapeutic benefit. And the key thing is that our PROTAC has to form a protein-protein interaction between the HDAC enzyme and the E3 ligand for proteasome-mediated degradation. Okay, so this is the molecule that we synthesized in my lab. This was actually synthesized by my PhD student, Josh Smalley, who was also an undergraduate at the University of Leicester and did a year in industry as well, and then stayed on with me to pursue his PhD. So this is the PROTAC that Josh synthesized in the chemistry lab. So this is the part that recognizes the HDAC enzyme. This is the linker part here. So you said we have to have a linker. And this is the ligand that recognizes the E3 ligand necessary for proteasome-mediated degradation. And here's a little bit of biology. So this is HDAC1, HDAC2, and HDAC3. Don't worry about the tubulin. And you can see, and this is the concentration of the PROTAC. And as the concentration of the PROTAC increases, HDAC1 is disappearing. So we can see here at 10 micromolar, it's completely gone. At 4 micromolar, there's only 36% of it left. And that's the same for HDAC2. HDAC3 is being reduced, but not as much. So we have developed the first molecule, PROTAC, that is capable of not only inhibiting a HDAC enzyme, which is important in cancer and other diseases, but degrading and removing those enzymes. Okay, and this is a model here. So this is the HDAC enzyme in orange, the protein HDAC enzyme, and this is the E3 ligand in purple, and this is our molecule here, which brings these two proteins together to form a protein-protein interaction. This red part is this part here. This is the linker going across, and this is the blue part here. So bringing these two proteins in proximity to induce degradation. And this is a zoomed up version of this. So this is looking at the molecule now itself. This is the ligand for the HDAC, and this is the linker, and this is the E3 ligand. We've just published this work in the journal Chemical Communications if you'd like to find out more information about it. And if you want even more information, you can email me at this website.